Hello, everybody. Welcome to the ISCB Student Council webinar series. And today I'm happy to have the opportunity to introduce our keynote speaker, Dr. Afshin Beheshti. Dr. Beheshti is a bioinformatician and principal investigator at NASA Ames Research Center. He's also the visiting researcher at Broad Institute and co-president of COVID-19 International Research Team. Today, he will be giving us a talk titled Science Driven by Space Biology Omics Data Utilizing NASA's GeneLab platform, which will highlight how NASA GeneLab has been critical in accelerating research related to space biology. Let's welcome our speaker, and I will request Dr. Beheshti to go ahead with his presentation.
Yeah. Oh, thank you so much for the introduction and the invitation to speak with it to everyone here. So I don't need to introduce myself since Shoni did a good job. So let me just dive into it. I'm going to give you some background on what NASA might be interested in, and then what in a sense what GeneLab is about and what happens. I mean, some of you might have bioinformatics background, so you might know all of this. Some of you don't. So we'll go over a little bit of it. So every 10 years, as the title says, NASA has a Decadal survey that's released. The next Decadal survey, of course, 2021 is now, is being put together now. And usually what the Decadal survey is that in NASA, anyone here could actually write a white paper for it, because it reaches out to the community to send in white papers to NASA, Decadal survey portal, and then they look at what the science from people around the world and people around the U.S. that what specific type of questions seem to be important, that they want to answer. So back in 2011, they had a Decadal survey, and the last one is here. And back then, they started realizing, of course, in 10 years ago, the technology for omics, like transcriptomics, genomics, and so on, proteomics has been substantially, increased, and the costs have significantly gone down, as you can see with this plot here from the NIH showing the sequencing costs, the genome costs. That was back in 2011, and what is an older graph is 2017, but 2021 it's about the same, as you guys probably know. So that's back then, 10 years ago, that was the thing that people started realizing. Oh, omics can be used as a powerful tool to explore biology in general, but specifically for space biology, what could be used to tackle some of the questions that we need to tackle. So GeneLab was born around here, and since then, as you know, the sequencing technology has gotten better and cheaper. Before we dive into what is GeneLab, not sure what the background of people out there, but I should describe what the space environment is and why you want to study space biology, and what components of it is important. So space environment itself and an astronaut in space is composed of several different things that, of course, we're not exposed to on Earth. For example, there's a lack of gravity. So it's microgravity events that could cause different types of health risks. And then there's space radiation, as you see from these plots. Two of them, one source is from solar particle events, or like the solar flares, as you can see here, which is a high dose of protons that someone could be exposed to in space. And then the other one is galactic cosmic rays, which is from anywhere in space, usually from supernovas or something like that, where heavy ions are dispersed through space and astronauts are exposed to them. So currently the ISS, which is in low Earth orbit, which is here, it has a lower dose or a lower impact of the deep space radiation because of our protection we get from the Van Allen belt, or the magnetosphere from Earth. That helps lower the dose and lower some of the protect you from some of the heavier ions that might be there. So the astronauts currently being doing experiments and going up in space are at the International Space Station in low Earth orbit. But as we know, there's future missions planned to go back to the moon, establish a colony there, and then eventually go to Mars. So this is kind of an estimate of doses compared what will happen when someone goes to the deep space mission to Mars, for example, which would take about a year, and compared to doses that you might get on Earth, for example, X-rays, and then compared to, this is an older plot here from the DOE, but the Skylab, which is the ISS, this is kind of the dose equivalent you get for about a few months if you spend about 3 months you spend on the ISS. So you can see you get a significant higher dose up there. Plus, the type of dose you get is different because there's heavy ions, which I'll get into in a second. So there's these two major components like space radiation and microgravity, which contributes to health risk. And there's other factors that that NASA considers a hazard is like the distance from Earth. For example, when you're on the ISS, if any emergency happens, someone could be an astronaut could easily come back to Earth within a few hours. While if you go to deep space mission to Mars, that's not the case. So they're on their own, and hopefully nothing will happen. But if something does, they have to be prepared. And the confinement is another kind of a psychological issue where you're stuck in a tube, for a long period of time, and and so on. And then so the whole goal of all this experiments and idea of space biology is make it safe enough for the astronaut that all the biological effects that we know of, which I'll get into in a second, can be mitigated by some sort of countermeasure or some sort of therapeutic to actually help the astronaut stay healthy for these long-term missions in deep space and in those hostile environments. So as I said, deep space radiation is composed of your solar particle events that happen like from solar flares, and then you got the galactic cosmic rays, which is anywhere in space, usually from something like a supernova might happen. So then you get these heavy ions like iron, helium, proton, and everything in between, being flung around in space, and then that can easily the astronaut can get irradiated by these heavier dosages, which we don't experience on Earth. So that's something that is unique to deep space radiation. And the low, as I said, the magnetosphere traps some of the radiation around it, so then you get a lower dose of it or a lower amount of heavy ions that the astronaut is exposed to. And then Earth, definitely, we're not exposed to that. So some of the health risks that astronauts are exposed to, there's a lot of health risks here. There's a review paper that people are interested in that was in Cell that we put out, talking about the fundamentals of biological features that are in space, and then also talk about the health risks that are here. As you can see, there's many different health risks associated, and common things between men and women, and and things that are unique to men and women too. But things related to, like, as you could imagine, bone loss and muscle degeneration because there's a lack of gravity, cancer, long-term cancer risks, there's cardiovascular issues, CNS issues, there's central nervous system issues. There's even things related with your eyes, which is the space-associated neuroocular syndrome, SANS, as it's called, abbreviated, which would affect how the astronaut's vision might change over time. So, and again, like sleep and things like that. So there's a lot of different health risks that are currently being studied by many investigators like myself and others that were involved in these studies. I'm going to present here, and then many others that we're trying to come up with good countermeasures to combat these effects. So again, an astronaut could be healthy and staying on Moon for a while, going to Mars. So then I just want to get into quickly what type of experiments that people do with space biology. So these type of experiments, which I'll get into in GeneLab in a second, end up eventually, like the data from these end up in GeneLab. So it's important to know what type of experiments are being done in space biology if you're not familiar. So one thing is, experiments are done in space, right? So these are our mighty mousestronauts. And this is a video from them on the ISS. So what they do is, people send mice in the payloads that go up to the ISS. And previously in the shuttle missions, when we used to have shuttle missions before the ISS, International Space Station, the mice were put on the shuttle. And then, they go on for a period of time, depending on experiment, in these cages that are designed specifically to go on the International Space Station. And then the mice, as you can see, eventually acclimate, as the astronauts do, and float around nicely in space. And then usually, at the end point, either the astronauts do some experiments or they're flown back to the Earth, and then the mice are then euthanized, and then the tissues are taken for omic type studies and other type of studies. So that's the type of experiments done in space. And we also send like cells in vitro, cells in a dish up in space, and then there's plants set up to space, microbes. So there's basically those type of experiments done. So there's also analog type of experiments that are done on Earth. And one type of analog to simulate space radiation, because you can't send everything to space and do all the experiments you want. There's only a limited amount of time and room you can do and experiments you can do. So we need to come up with ways that we could try to simulate what occurs in space. And more importantly, as I said, the International Space Station is around the low Earth orbit. So we have no way physically to simulate what's currently, in deep space outside of our magnetosphere until we say we have a moon base. But so in that case, we also have to try to simulate what occurs outside of the low Earth orbit, the deep space radiation. So then to do that, there's experiments we do at Brookhaven National Lab, which is a high energy physics collider there. What they have done, NASA had set up the NASA Space Radiation Laboratory, which was probably around 20 plus years ago, that they basically said the only way that you could actually capture one of those heavy ions to, let's say, irradiate a mouse or a cell or something else is to use these high energy colliders. And then when these high energy colliders are creating these heavy ions, then you could take like an iron particle or a helium particle or oxygen and so on, and take one and then take it into what's now this building, the NASA NSRL, NASA Space Radiation Laboratory, where the beam is here. And you can put samples in front of the beam and irradiate, let's say, a group of mice or cells or whatever your experiment is about. And then you could, with the heavy ions that are there. So that's one way you could do deep space radiation experiments. And really, Brookhaven National Lab is the only place in the world that you could do these types of experiments with any type of heavy ions you want, or a combination of heavy ions that would simulate like the heavy ions mixture you might get exposed to when you're exposed to those galactic cosmic rays. So that's one type of experiment that people can do. And for this, of course, you would apply for NASA grants and get your NASA solicitation, hopefully awarded. And then, in your grant, you say, I want to irradiate something and do these experiments. And then you end up going to Brookhaven for a period of time, setting up your experiment and running it. And then hopefully then you get some really interesting results and you get omics data that then eventually you can deposit on GeneLab. Another type of experiment is like for microgravity. There's simulated type of experiments you can do with mice analog. So one of it is these hind limb unloading, as you can see. So the hind limb unloading is where you suspend the mouse like this by the tail for a few weeks, so that then they can't use their back legs. So that simulates the unloading or of the weight in the back legs in a sense. So then when you have a lack of gravity, is the same idea. And you can see that there's some simulation in the muscle tissues that are similar to what occurs in astronauts. And another model developed by Stuart Rudkov and Miriam True at Beth Israel and Harvard is partial weight-bearing rat model, where they have these kind of harnesses that you could put rats at different elevations to simulate different types of gravity, like from 20% gravity, which could be on the moon, to 100% gravity on Earth, and then everything in between, and then 0%, like the tail vein here. So the hind limb setup here, to the setup here. So there's different models that they've developed that's really nice. And then of course there's human analog studies, which I'm not showing here, but there's things called bed rest studies where they get volunteers to actually lay in a bed at a 6-degree tilt angle in bed for about 30 days. So that's sort of simulating like the lack of muscle use. And when you're in space, you got fluid shifts in your head that with that 6-degree angle when you're lying in bed would kind of simulate that. And then the volunteers would let you take samples like blood samples or other types of less invasive samples out of them so you can run omics on. So those are the type of experiments you can do. So what happens when you actually generate the data? So let's say you generate all this omics data and you analyze it yourselves, and then you want to share it with the public. This is where GeneLab comes into play. So GeneLab is sort of was set up in a sense to first to be a data repository for any type of omics that's out there. And what the infrastructure and the idea is, we have a lot of genetic diversity. As I said, there's a lot of different types of experiments you can do. And I mentioned the rodents, but they've also, a lot of people do experiments with the fruit flies, Drosophila, C. elegans, which are flown to space or done here on Earth. They've even done experiments with fish, like zebrafish, things like that. So we got a lot of diversity that simulates different types of aspects of human physiology. For example, the C. elegans are good for muscle type of one thing is good for muscle. There's many different things you can use it to. The fruit flies is also for like oxidative stress, things like that, so on. So every animal model has its unique capability to be associated with something in human physiology, the fruit fly studies, and then how it relates with humans. And then there's of course microbes. So there's microbes that people look at the host and the gut microbe changes, but then also there's the environmental microbes that grow in the ISS, because you can imagine a tube that's there, and microbes accumulate around things. So they want to know what kind of bacteria is accumulating from actually the existence of humans in there, because previously when it was sent up, it was microbe free. And then over the last 10, 15 years in the ISS, then, from the microbes that are shed from humans, things eventually, as you would in your house or anywhere else, if you don't clean, you clean as well as you can, but, there's still microbes that would grow. So, people do studies on that and end up on GeoBank. And then plants. So, people do a lot of studies, space biology studies on plants, where they look at, how to do basically the idea would be for long-term mission, how can you grow a sustainable food supply on, let's say, a mission to Mars? Then you won't have to store as much food and bring it, and you can just grow your own plants in space. So those omics data from that ends up on GeneLab also. And then, there's ways that you can use multi-omics approaches because it's not only transcriptomic data for genes, there's genomic data, there's methylation, like for DNA, epigenetic data. And then there's also proteomic data we have on there, and metabolomic data for metabolites, metabolism type factors. So all that ends up there. And is there any kind of multi-omic kind of approaches that can relate all these different species and type of techniques together? What is GeneLab set up as? So we have a submission portal where a PI or one of you bright students here can have data and then submit it. So it's kind of a portal where you set up that it's kind of set up where the user can, and then of course people in GeneLab who are involved with this, if there's any issues, they could always help you, set it up and submit your data to GeneLab. So then as I said, it's usually a lot of the experiments from a PI or missions experiments, and then there's some samples that are actually processed by GeneLab. And usually the PI experiments, if you have, let's say, a grant by NASA, NASA usually give you a year grace period time from the time you generated your omics data to actually get your own papers out first before you submit to GeneLab. But when GeneLab processes the data, we're not a PI or anything. So we basically, at the moment we're done processing, it's right on GeneLab, so anyone can jump first on that data set and process that data and come up with a novel idea and a paper from that. So, it's always good to keep an eye on GeneLab if you haven't, because new data sets that are processed by GeneLab end up on there that no one has touched yet. So the whole is open to the entire world now that, you could end up coming up with a novel, nice story and come up with a paper and show something neat that's happening in space biology. And then so we got this data portal. As I said, we got the raw data, which is basically the raw data that maybe the hardcore bioinformaticians, computational biologists want to get their hands on. We got the process data, which was agreed with pipelines that are agreed on by the scientific community, which I'll get into in a second. And then we got metadata on there, which is, I'll get into this also, which is basically kind of environmental type data, like the radiation dose absorbed, let's say in space or on Earth in those analog studies, or temperature, things like that. So the process data itself, which I'll get into, is basically you don't have to process anything yourself, and you can use what the pipelines we use, which this is all transparent on GeneLab. And so anyone who, let's say, is not as computationally inclined and you just want to take the process data, look at the list of genes and run their own things and look at what important questions they have. We have also an analysis toolshed on there through the Galaxy platform, if people are familiar with that. But basically, it's a pretty easy to use platform where you can plug into GeneLab right away and get a data set and analyze things. It's kind of a window type based analysis tool, which we also have a tutorial to use it. And again, the environmental portal, as I said, is really the metadata, which we have all the information on, like temperature, radiation, absorbed dose, and so on. And then we have also a visualization portal, which could easily visualize the data, which I'll get into in a second. And then, of course, as I said earlier, we got pipelines and interactive tutorials, and all this stuff will eventually lead to new knowledge, which I'll show you some examples of how that occurs. So the data itself, democratization, as I say, is how the data works. We have the raw data, as I said, it's basically mapped reads for transcriptomic data or raw proteomic data. And then we got the differential expression data, which is basically, the raw data is basically for like the hardcore, as I said, bioinformaticians, data scientists. Once you get to the higher data, the differential expression data, like the fold change values or things that are more processed, I would consider like the biologists that are not these computationally with the computational background, but just want to get the process data and, as I said, plug away. And then we got also like pathway changes and disease states, which as we consider as like citizen scientists, basically the average person who doesn't have a science background, like everyone listening here, or maybe some people are both citizen scientists who are listening here. That anywhere could be from like high school students to people who are senior citizens who are just have a hobby of space and just want to answer an interesting question that they're come up with. Like they read something and they want to say, is this gene being regulated due to these conditions in space? Does GeneLab have the data? Yes. Okay, good. They have the data. Now they'll look at it and then they'll come up with a neat, answer for themselves, which they could actually, work and find someone and publish a paper with. Within each of these groups, we actually have this thing called an analysis working group, which I'll get into, and an educational working group for the citizen scientists. So the analysis working group, which is here, is comprised of basically anyone here can actually volunteer and join, from students to even citizen scientists. And it's right now like 120 plus scientists. Probably it's a little more than this. I have to update this. But analysis working groups are comprised of basically scientists from all around the world that volunteer to help GeneLab. It was first set up to help GeneLab, come up with a consensus pipeline to, for those high order process data. Since there's many different ways you can analyze, let's say transcriptomics, proteomics data, it's good to have the consensus from all the experts in the that are there. And if these people agree, then hopefully the scientific community with our process data will be happy with it. But in the process, we also have these people then collaborating together and coming up with interesting questions for space biology and coming up with nice papers that I'll show you in a second. So then we have 4 groups. One is focused purely on different animal type studies and I lead the multi-omics systems biology group, which these guys are probably the more computationally inclined. As I said, that they and the one paper that I'm going to talk about, or the 2 papers I'm talking about here in this talk, is actually comprised of people from the multi-omics group. There's the plants group. If you have a plants background and want to dive into space biology, you can do that. And the microbes group. So these guys are focused on the microbes I described. And I said, anyone around the world, any level of career that they're at, whether they're undergrads, high school, graduate or higher, director, postdoc, anyone can join and be part of our groups. And depending on your interests, you can be as involved as you want or be involved a little bit with the projects. And some interesting papers actually come out with people collaborating. And you develop new collaborations with people who are interested in space biology. Go to this link here and you can join. Or if you just go to the GeneLab link, genelab.nasa.gov, you'll find the AWG information. And then there's another thing called educational working group, which consists of like educators from high school teachers to scientists from colleges and so on, that are starting to promote GeneLab as a way for teaching tool for omics. So currently we have there's an intern program every summer before COVID, when students can actually come, they'll come in Ames for a period of time, work on some interesting things with projects with actually GeneLab, from undergrads to grads to postdocs. And then now virtually it's opened up for interest. So you could always contact me or others in GeneLab to see about the internship opportunities that are there. And then also at San Jose University, the people started to do a curriculum integrating GeneLab for the bioinformatics tool bioinformatics a resource for actually teaching bioinformatics. So, things like this could actually be used for education and working and ways to promote, education about bioinformatics. And GeneLab has good data sets and good ideas to promote that. So if people are interested in that, you can always reach out. As I said, now GeneLab's been growing since our beginning in conception at 2014. And now we have, as you can imagine, we're not like compared to, let's say, the Cancer Genome Atlas, which has a lot of data sets coming in, because space biology is more of a limited area of research and there's limited things you can do. But that said, we have about 350 data sets now, and they're comprised about 50-50 about space, those spaceflight studies I described earlier, and then about ground. There's actually more spaceflight happening now, and it's growing more spaceflight. And as I said, the organisms that are being studied is almost everything. For humans, it's not actually human samples, but it's actually in vitro, so cells, human cells that are either sent to space or irradiated, those analog experiments in the ground. And the reason behind that is Johnson Space Center, the Human Research Program, takes care of the human aspect of it. As you can imagine, there's still issues about if you do provide sequencing data from humans on the GeneLab platform, even though you de-identify it, it's for a clever person who's computationally inclined from sequencing, you can imagine there's only 8 or 9 astronauts at a time up there for a mission. You might easily be able to identify that one astronaut, from that 8 in that pool, as opposed to, let's say, a larger clinical study on Earth. You do for cancer, you got hundreds of patients, and when you de-identify it, you can share that, let's say, on portals like TCGA and have no concern about identifying someone. So that's still in the works, and hopefully eventually we'll get human data like that on GeneLab. And then we got all sorts of types of data. From the majority of it's transcriptomic. Previously, The legacy data was microarray from those older studies that people have done, but this is quickly, of course, over time changing as more and more people do RNA sequencing. And then if people are familiar with this, GeneLab is the most FAIR for any of these life science repositories, and FAIR stands for findable, accessible, interoperable, and reusable, which is a standard that has been set in place to make sure like data repositories and data platforms like ours actually will meet the highest quality and standard for data and being shared with the public and the accessibility. So people, haven't already gone on the GeneLab platform. This is the homepage, and the homepage, of course, changes over. The pictures and the news change. But as I said, there's the data repository link here, which you could actually just go search for the data. There's that collaborative workspace where you can share and organize files with direct access to the GeneLab files and the data there. This is our analysis little link where you can actually go and it brings you to a Galaxy type platform if you're familiar with Galaxy. If you're not, it's just a kind of a web-based platform to analyze omics data. And then here's your submission portal data, which you submit your data, environmental and visualization data. So here's the environmental example of that, where you can see we got many different things you can get, and this is one of the dosimetries from different missions and different GeneLab dataset. GLDS stands for GeneLab dataset, if you're not familiar with that. But different missions and different types of data you get with relate that. You can relate to the analysis you do from the omics. The visualization right now we had developed earlier on standard visualization from each data set where you can look at, for example, PCA, principal component analysis plots, volcano plots, or heat maps of that data set quickly. You could also, look at different correlated data and look at different genes, but we're also now going data visualize 2.0 where the previous visualization only did single viewpoints of each data set. But now we're with visualization 2.0, which is coming very soon. We can actually then do cross comparisons and data comparisons between multiple data sets on there. So that's what we're going to have, like an interactive network, and some other neat features that people can play around with, and also like pathway enrichment data that you can look at from each data set. So there's a lot of new data. So I mean, a lot of data we've already analyzed for people. So you don't even have to do too much work if you want to answer your interesting question. Just download the data and put a paper out. Okay, so now that I'm familiar with GeneLab, now I'm going to get into the actual how you can use GeneLab and what papers have come out and novel scientific theories and how GeneLab can be used. So one paper I'm going to discuss is a paper that came out of our analysis working group, the multi-omics group. It was a collaboration. It was a long, well, it wasn't that long, but it was a good haul, where it was a lot of work and many people involved. So this paper had about 30 some authors on there. All of them, most of them were from the analysis working group, so I'll get into in a second. And so this was actually part of a larger package where I'll talk about in the paper where me and another principal investigator, Chris Mason from Cornell Medicine, and another principal investigator, Susan Bailey from Colorado State, we put together with Cell Press a large space biology package, which the landing page is here, the biology of spaceflight, which included 29 different papers, and some papers from me, some papers from and a lot of papers from other PIs and NASA investigators internationally too, from space biology investigators. And one of the papers that Cell highlighted that ended up in Cell is this paper, a comprehensive multi-omics analysis reveals mitochondrial stress, which I'm discussed right now, and is like a central hub. And if you don't know, the mitochondrial is sort of the cell's powerhouse, right? As this guy here, the mitochondrial guy is here. So, in a sense, all your bioenergetics is run by the mitochondria, right? So if you want to be healthy and have a longer life, you want to have a healthy mitochondria. And usually as you grow older, your mitochondrial is going to get suppressed more and more just due to naturally of age. So, if you have a health, and exercise usually helps boost your bioenergetics in this case, or, eating healthy and things like that. But there are some, health conditions like diabetes, heart condition. The more obese you are, the less your mitochondria will be behave properly. So, there's other kinds of factors that would decrease your mitochondrial ability to work properly. And to keep in mind is that, for your mitochondria, that astronauts are probably the 0.001% healthiest of the population. So their mitochondria is probably very healthy and very, strong. So, that's the kind of population we're studying here. And you can imagine, what I'm going to show you here, this function that occurs in spaceflight from the astronauts. If it occurs to them, the average person like me, going to Earth, going outside of Earth, I'll be probably a lot harder hit by the effects of space. So the people involved, as I said, there's a lot of people involved. It's all from the majority of them are from the analysis working groups, and then some people were brought in as collaborators that people knew. And for example, all these people from Johnson Space Center, Scott Smith, Brian Christian, Sarah are from Johnson. They're part of the AWG, but they're from collaborators that are within our AWG that we brought them in to help us out, which I'll go into what they've done. And same with Doug Wallace. He's not part of the AWG, but he was a collaborator with people here that we brought him in. And of course, Doug Wallace is the founder of mitochondrial genetics, so it's great to have him here to help us guide us to the mitochondrial story that I'm about to tell you. So a big overview of the data used from GeneLab. So we were trying to, the main question we're trying to ask is, initially when we started, I proposed could there be a master switch in space that causes all those systemic effects that I previously had talked about to occur? So, or is everything kind of in its own component, in your body? Like for example, there's liver issues or cardiovascular issues. Are they related? Or is this going to be, in my opinion, I have a physics background and I switched over from physics to biology. So a physicist tries to simplify things. So there's always a kind of a systems approach that way, hence systems biology, where we try to relate, that we always think there might be a connectivity, let's put it that way, where, when you get that whole insult in space, that the dysfunction that might occur in the heart should be related to the other organs, because as you can imagine, we're thinking of the system as a whole. So to tackle this question, since I already said GeneLab has many different platforms, you got to be creative. And one thing with GeneLab, the more creative you are, the more interesting you get really neat results and novel findings out of it. So what we did was we tackled a few different data sets from mice that were flown in space that are available in GeneLab that you guys can go, transcriptomic data. And we looked at many different organs, and these are all these numbers, all these different organs from the mice. We also looked at cells, these type of cell types that were flown to space, and analyzed that. And we not only spent time looking at the transcriptomic, the genes, we also looked at proteomic data that's available in GeneLab, methylation data that's available in GeneLab, and metabolites or metabolomic data that wasn't available in GeneLab because the collaborator was actually who provided the data was running it, but now is because they dumped it on GeneLab, and that data is now available on GeneLab for people to look at. That's in this paper. And then using all the creativity here and all the brain power that's backing here from many different backgrounds and disciplines. People did many different types of analysis independently to look at what's occurring in each of these, conditions, organs, conditions, and so on. And what was interesting is, since they went independently by themselves, we came up with a common theme that was popping up, which I'm going to get into, which is mitochondria. And then the last thing we did was take we went to our collaborators at Scott Smith and Brian Christian were at Johnson Space Center, and that's Johnson Space Center, and they basically had physiological data from astronauts from the last 20 years, so they said, so we gave them a list of factors related to mitochondrial dysfunction that we're seeing occurring. And we said, would you have possible human physiological data that you could provide us, and to maybe say, if these factors are occurring based on omics, can this be occurring with the physiological changes that we expected to see, what happened? So they were able to provide 59 astronaut data compiled together, physiological data. This is like the largest amount of astronaut data to appear in one paper. And lastly, we were able to provide the twin study data. That's, if you guys are not familiar, this is a study that with the NASA astronauts, the twins, Scott Smith, who went in space the longest duration for a year, and his brother Mark Smith, who was an astronaut also, but at the time, wasn't in space. He was on the ground. So they compared those two. It's an N of 1, but at the same time, it's the only type of unique data we have where you take samples from these astronauts that were, one, exposed to space environment for the longest period of time, one that was on the ground. So we have that data, and the PI that says was is Chris Mason, who, he's part of this study and analysis workers, and he provided the data that we could use for this paper too, to help further confirm in humans what we see in the rodents and cells. So this is a big global profile and overview of what's occurring. We see that, that in general, when you look at the genes, it's transcriptomic data of all the different tissues, the metabolites or metabolomic data or proteomic data here, that, there's different overall global regulations happening. Like you see, for example, the average this is up regulated or down regulated. Muscle type tissues tend to be highest in the list. And you can imagine that because due to microgravity, that makes sense. And then there's other things like liver dysfunction comes down, things like that. And like for methylation data, you can see liver up, and again, muscle type tissues that are being up. So that's kind of the global profile. But then once you start getting into actually the biology and the nitty gritty of it, what we see is, and I said, when everyone went off on their own, analyzed each of these data sets on their own, then we came back and said, what did you see was a key factor? Let's say in the liver. And what someone said is mitochondria is the top has being regulated. Someone else came back and said, it's the kidney, mitochondrial. And what eventually came back, even like, for example, the eye, you saw pathways related to the mitochondrial, which if people looked at them independently, someone, let's say for this eye data or the carotid artery data, which we have, might have thought doesn't make sense. But then when you look at the system as a whole, you piece the puzzles together now, right? And then you see that, mitochondrial is a key factor being changed commonly no matter which tissue you look at. And this is what this big network plot is showing, where whether it's the proteomic data, which is the circles in the middle, or the transcriptomic data, this is the circles outlier, and depending on the tissues that are going up and down, mitochondrial functions occurring. And this is even the case with cells in a dish outside of an organism. The top 4 pathways that are being commonly regulated between these is all related to mitochondrial related pathways, which is extremely interesting because now you took cells, these are human cells outside of an organism in a dish, and you might expect different response, but again, mitochondrial is a common theme popping up. And the interesting part is that, it's not the mitochondrial dysfunction in the organs are not necessarily universal. For example, they all might not be going the same direction in the liver compared to the muscle. But in a second, I'm going to explain why that is, because that makes sense there. Whether it's methylation, omics data, whether it's in the muscle or the different organs here, same way. This is kind of like an overall profile of mitochondrial specific genes. There's a resource on Broad Institute site called Mitocarda, which it's updated periodically, where it's a good resource if you just want to download a curated set of what is thought to know and be associated with mitochondrial genes. They have a list, and that list is about 1,000 some genes that are thought to be all mitochondrial related. It might not be, and as people discover more and more, the list is updated. So when you overlap that mitochondria or the genes that are related with that thought to be related with mitochondrial to the overall regulations happening here, you can see, for example, the liver has the most related percentage of genes that are being regulated in the mice when they're flown in space. And then for the muscles, you can see that, the soleus muscle, which is one of the muscles that people know is heavily impacted by microgravity. You can see, again, mitochondrial heavily impacts that too. And again, all the organs are, but you can find key factors. Now, when you look at, let me zoom into this top plot here so you can see it better, but you look at the metabolites in the muscles, and again, mitochondrial, these blue fonts as it relates to the mitochondrial factors are the ones that are really associated with mitochondrial, and again, pops up again. And then when you look at mitochondrial specific genes, for example, the integrative stress response genes, which are related with regulating mitochondrial and stress response, there's some interesting patterns happening where some are repressed in some organs and some are upregulated in other organs. For example, the muscle here is upregulated, while in some of the other muscles it's downregulated. Again, that relates to the location of the muscle and how some places are overcompensating for the other places. Oxidative phosphorylated genes are related to mitochondrial, which, as we know, when you get mitochondrial stress, it causes like reactive oxygen species, and it causes oxidative phosphorylation to occur because of the bioenergetics are not working properly. So that's why we see that occurring. We got a little Homer Simpson here trying to say, I tried to see the importance of mitochondrial, but then of course you lose energy, and that's really the oxidative phosphorylation. And then what's interesting, now you look at the human data, And this is the twin study data. People ran PCR. If you're not familiar with PCR, this is when you run the polymerase chain reaction for the specific gene you're looking at, and then see what happens. So this is the grounds. This is Mark Kelly here for here, whatever is a TW, or sorry, whatever is HR, and then the Scott Kelly is a TW, whatever is the he flew in space for. And you can see these are mitochondrial nuclear genes that are specifically being upregulated while this does not occur for Mark Kelly as a ground. Luckily, when Scott Kelly comes back, it seems like the mitochondrial functions started to restore back to normal. As you see, you got during flight and post-flight is the orange, and not all of them come back down, to the basal level before, but and maybe if you had more time points afterwards, maybe they will. But luckily, some of them do come back. And then this is the astronaut physiological data from those 59 astronauts that were compiled from the shuttle mission to ISS, and you can see factors related with oxidative stress, which is here, is being suppressed. These urinary markers are actually related also with oxidative stress, and they're being upregulated as we've shown with the transcriptomic data. So this indeed, and this is pretty significantly higher regulated when the astronauts are in space. So these are before flight, FD is when they're during flight at the different time points, and then when they return is 0. So luckily, these markers tend to go back to the normal levels when the astronauts come back, which is good to see. And then what's interesting here, you can do some really nice, clever analysis, which is done. This is done by Deanne Taylor's group and Jemmy Mann at Children's Hospital of Philadelphia. It's part of the analysis working group. So they did some clever analysis where you can use transcriptomic data to do metabolic flux analysis. I won't go into the details of this, but what they do is basically take the transcriptomic data and predict metabolites, how the functions change. And what they saw is here, for example, this is the liver. They see that indeed for mice that were flown in space, you got mitochondrial related functions being upregulated while mice on the ground were not, which is here. But when you look at, and again, you look at oxidative stress thing, phosphorylation, which is being suppressed, this is for the muscle, but the liver related to being upregulated. But the interesting part is that when you look at the muscle data, which is here, you see the opposite thing happen. And we know for muscle wasting literature that indeed, when the muscle is wasting, that you do see suppression of mitochondrial, like oxidative phosphorylation happening, being suppressed, or being in the spaceflight samples, as the mitochondrial related things are being suppressed. And we know from muscle generation literature that indeed, when muscle generation occurs, your mitochondrial is suppressed in your muscle, while other organs like the liver are trying to overcompensate for that and produce more and more mitochondria to try to help you not have your muscle waste as much. But unfortunately, the muscle wasting here basically overpowers the fact that the liver is trying to overproduce that and trying to help your body get your bioenergetics back to a level where it can, but never does. So that's what earlier I said, not in every organ, the mitochondrial doesn't actually go the same way. And then this is from the NASA twin study data. This is data from Scott and Mark. And we see that, and these are the different cell components, cell types that through Chris Mason's lab we analyzed. And this is looking at the mitochondrial specific genes. And what's interesting is that you can see that here is the different cell types, and this is from pre-flight versus the ground, in-flight, and in-flight, post-flight versus the ground, and then comparing Scott Kelly to himself over time when he was in flight. And you can see that the mitochondrial genes have a shift in the cell type composition and where they go. And this gives you some insight how mitochondrial changes occurred in Scott over time from, comparing this pre-flight to ground, flight and ground, and then, and so on, and how the mitochondrial shifts, which is something that doesn't happen in Mark Kelly. So there's definitely a lot of mitochondrial changes happening, in Scott Kelly in space. Now there's universal downstream effects that occur and dysfunction that occurs. So, what's interesting is that previously people knew, like, for example, immune suppression, immune dysfunction happens. And they were focused on that. But what we do know about mitochondria from all the mitochondrial literature that happens in the clinic is that that's all upstream of that. So when mitochondrial dysfunction happens, it will cause immune dysfunction and downstream things. And this is exactly what we see here. I'm getting probably close to the end of my time, so I'm just going to go over this part quickly, and then you guys could ask questions. But yeah, so we see that basically, again, mitochondria, this is like the astronaut physiological data with different immune factors from vitamin D to VEGF to inflammatory factors, that indeed we see that, for example, when the astronauts are in space, inflammatory factors are heavily increased. Same with like VEGF, but vitamin D with suppression is related to mitochondrial dysfunction and immune dysfunction. When that's suppressed in your body, you want that up. That's why people get vitamin D supplements. Again, another consequence of mitochondrial dysfunction is leading to lipid and fatty acid dysregulation. And if you're asking what would that happen if, let's say your fatty acid dysfunction is like increased, which is what we see here in the liver. Diseases such as non-alcoholic fatty liver disease, which would happen either, let's say, if you drink too much alcohol, or from other kinds of liver diseases would occur where lipid accumulation happens in the liver, and eventually your liver would stop working. So this is what we see happening here in the liver tissues. This is the liver tissues here, this is green ones. We see a lot of factors related with lipid accumulation, fatty acid dysregulation from this metabolic flux analysis that Deanne and her lab did. We also see physiologically cholesterol levels are up in the astronauts when they're in space. And then when they return, it doesn't really go back down. It's going back down, but still significantly up from before they left. Again, this is the omics data from the transcriptomic and the proteomic data, and it shows the same thing. But again, when you look at the muscle is degenerating. So you see the reverse happening, just like the other mitochondrial dysfunction, where lipid and fatty acid dysregulation is being suppressed as opposed to increased, which makes sense again with muscle degeneration in the literature. And then other downstream factors of mitochondria we saw happening is really a cell cycle. So you want your cells to be dividing and reproducing in a good, healthy way to keep from causing mutations in cells that could lead to different diseases like cancer. But we do see factors of cell cycle, which is again is a consequence of mitochondrial dysfunction. Everything leads back to the mitochondrial in this case. Where you can see that it's being heavily regulated, whether you're looking at the in vitro data, that the cells flown in space, or the mice. There's also things related with like photoreceptors, which can be related to your eyes being changes happening, and also circadian rhythm issues. So we see again methylation data, circadian rhythm. And we know in astronauts when they go in space, even though everything's done, that they would get their sleep cycle regulated. Their circadian rhythm issues, there's always happening because due to the microgravity that's up there, it causes issues. So, that's an issue we know of, but again, mitochondrial is one of those things that are upstream of this. And we see here connecting it to all the circadian issues that are happening downstream. And another interesting part is the olfactory pathways pop up, being upregulated. And, as we know from the COVID era, right, people know that you lose the sense of smell and taste, like when you get infected with the virus and your olfactory issues occur. Well, astronauts in space have reported that when they go in space, their sense of smell and taste also get sort of impacted and change. Whether it's permanent or not, some of them come back, they're fine. But we see this happening here. And again, that's a downstream effect of mitochondrial dysfunction. And I won't go on the Renin thing due to the sense of lack of time. So in summary of this work is, basically, if you go in space, you might. What we're saying from this paper is that, it's really mitochondrial is driving all this dysfunction that causes all the health effects we see downstream, or one of the main drivers. So now the idea is, well, there's a lot of in the clinic, there's a lot of studies being done with potential mitochondrial therapeutics that you can use for mitochondrial diseases. You can easily now apply those kind of therapies and test those in astronauts and see how effective they'll be, since people know there's some effectiveness to these therapeutics that are being used to cover some of these features that are happening in space and fundamentals of biology. You got about 10 minutes, so I won't go over this other project, but there's many more projects. This is a project that you guys could read the paper about. Where's the paper? Let me go back. Sorry. There's many different papers that people at GeneLab. It's another paper that we utilize GeneLab as a way to answer a question about could be a universal response for space radiation from the ground studies that are done, that I mentioned, to the spaceflight studies. So, using some clever ways of looking at all these different tissues that are there and using the ground analog, comparing to the, ISS missions, we're able to use like pathway analysis tools to actually come up with a nice and clustering methods and bioinformatic tools and modeling to actually come up with some sort of a fit and some sort of way we could actually come up with pathways to predict what could occur over time in deep space. And I'm going to go skip all this to leave room for questions. But one main part of this was these two studies were done independently. And this paper came out earlier in January 2020. The mitochondrial paper came out in November 2020. So very different. So independent groups were working on this, and I was involved in both. But one factor which is interesting, we do see mitochondrial passes and purely independent, purely driven, unbiased by using GeneLab with mathematical tools to come up with an unbiased way to predict things. And again, we see mitochondrial pathways being independently heavily upregulated in dysfunction occurring as an increase of dose. So that's a bit the idea of this paper, and I'm just going to skip over all this since low on time. And for this paper, these are the people who are involved. So you can see some overlap with the other papers. So in summary, I hope you see the usefulness of GeneLab, that you could actually come in, use the samples that are being processed there, that are constantly being updated, and more samples and data sets are being, from the 350 we have, more and more keeps growing to newer data sets. And the new knowledge produced, you can see some high impact papers actually can come out of what's there. So, and then the last plug is, the paper that I discussed today is not the only paper that's out there on space biology. There's lots of space biology papers that not only use GeneLab, but used, their own type of research. So if you actually, again, go to the biology of spaceflight on Cell Press, you can find a good list of papers, about 29 papers, to dive into space biology type research and see what people have done. And just a little plug, these are we got Cell Press decided to provide the covers for back in November, and you can see these are the pictures Scott Kelly, they picked from Scott Kelly that he took when he was in the ISS, different nice pictures that he took that Cell used for their covers. So, and of course, this is the GeneLab team that helped develop GeneLab, and this is the older team. This is the newest team that help maintain and keep GeneLab going, so people can actually use it for coming up with new and novel ideas. So, okay, I think that's covers everything for now, and I'll stop sharing my screen so you can ask questions.
Thank you, Dr. Behesti, for this wonderful presentation. So I will look forward to questions from people. So I think there's one comment.
Oh yeah, okay, I read it. Okay, yeah, I see it. Yep. Okay, so hi, Afshin. It's really awesome that all the data is publicly accessible. Sometimes when I try to read papers based on data, they're behind a paywall. Oh, okay, yeah. Yeah, so that's always the issue too. So yeah, so if people are not familiar with this, some journals, well, unless you pay for open access in a journal, and your institution might, and your institution usually has licenses to get those open, those private, non-open access papers, but not for every journal. And I run into the same situation as you do. So unfortunately, I mean, we've tried. I mean, so whenever, if it's a GeneLab paper, we always make sure that the paper is open source. So we pay for it and make sure, we pay that extra, what is it, $1,000, $2,000. We say, we're going to make it open and make it so anyone can access that paper. But unfortunately, we can't make the PIs do that. It's up to them. So the other principal investigators do space biology, so it's really up to them. And we encourage people to make their papers, pay that extra amount, because I think it's well worth it. As you point out, you try to read the original paper, and you want to know what the person did that that ran that original data set. That's not us in GeneLab. Then it's difficult to get that. So there's really no solution around it other than some journals have a like a year embargo window, and then eventually the paper becomes open to the public and open, but some journals never do. So unfortunately, there's not a solution to that yet, but we always try to encourage people who deposit data in their papers, make it open, because that's the only way science gets pushed forward. Don't make things closed, but and be very collaborative. Let's see. Okay, so next one. Okay, so from Max, said thanks for NASA talk. I think it's really cool. The data is so open, and that you mentioned citizen scientists. Yep, I wondered whether you actually see and have examples of scientists using the data. Yeah, I mean, there's no papers yet, but we have the this program called, it's probably like the educational part. We have GeneLab for high school students. And that happens, that's run by a couple of GeneLab people, like Liz Blabber, who's at RPI now. He used to be at Ames. And then I think Sigrid Reich at Ames, and then a few high school teachers also help out. So, and then basically that's, people from the program used to be high school, before COVID, high school kids would come to Ames for a period of time, and then they would work with people there in person, but now it's virtual. So then it's now opened up to more, people who don't have the travel funds to come or not close to the Bay Area, they could virtually come. So we consider those kids citizen scientists. And then they actually come up with some really neat things. And then neat data. They're really smart kids that they could really tackle some scientific data working with us, with the people not me, but the people running that GeneLab for high school program. And then with the meeting every year that highlights this kind of work is the American Society of Space Biology. And it happens once a year on November. And usually the kids who can do it would go there and present a poster, their work. And they do some really, sometimes better work than the rest of us do in the scientific community. Highly encouraged, for people to participate. And if you know any high school kids interested in space, tackle it. Yeah.
So any more questions, guys?
Oh, and I just want to make one plug. I see Stephen Keegan had mentioned the multi-omics group. Yeah, so if anyone wants to join the analysis working groups, whether it's the one I lead or any of the other ones, you can reach out to me or go on GeneLab, just send an email. The email list there, and anyone here, or anyone that wants to join, forward it and tell them to join. More the merrier.
So I have one question here. So our international students can apply for these working groups, or it's specifically for people working in
No,
US?
it's anyone. Yeah. So we right now have, I see in my group, I have people from Europe. There's even people from Australia and Taiwan. Wow. They're hard in New Zealand. We entered someone, and of course, it's people from the other side of the world. It's a little harder for them to join I have monthly meetings like every Wednesday, third Wednesday of every month at 11 a.m. Eastern time. And, people on the other side of the world, of course, they can't join because it's in the middle of the night, but sometimes some of them do.
Okay.
But usually we record the meetings too. If someone, let's say from Taiwan or China or Japan wanted to join, right, they could hop on and join and then see the recording and then email interactions, stuff like that. So yeah, there's no restrictions other than, you have to be interested in space biology.
Okay, I think we have one. So I had one technical question. So you were talking about the changes in the mitochondrial genome. So what was the conclusion of that entire study? Like, is there any time duration that the astronauts can only be up till that time, or what was the conclusion of that study?
So yeah, unfortunately we didn't have the time, like other than the Scott Kelly data, right? He had the time points. Usually for the mice data, it's the endpoint we get is the time where we sacrifice the mice. And there's usually a couple of time points you have, whether the mice are returned back to Earth or the mice were on the ISS. And there's a few time points within them. So it's harder to pinpoint exactly when the time point happens when the mitochondrial might be dysfunction might occur.
Okay.
And as you know from Scott Kelly, it's N of 1. So but it's the best we have, and the only unique data set. So we could say, I would say, the mitochondrial dysfunction doesn't occur right away. It would probably take a month or so, probably the accumulation of that insult to injury, will occur over to actually make all those downstream effects happen. But that's some study that'll be really interesting to do. And let's say if you have a clever way of using GeneLab to come up with the time course, I think you dive into it. That's definitely a question we should tackle.
Okay. Any more questions? I think we have a comment from
the room. Yeah. Thanks. See your comments. Yeah. Yeah. And people email me too if you can share my email, and people have questions, they can always email me offline.
Sure. Yeah, a lot of people actually watch our the recorded sessions. Like we generally see that they are like 100 views by next day.
Oh, that's nice. Okay.
So yeah. That was a really wonderful presentation. We got there were a lot of data and a lot of information, and I hope like a lot of people who has has been there today will be able to, maybe join in the working groups as well.
Yeah,
Thank you
yeah,
so much, for your time. And yeah,
yeah. No, thanks for having me. It was fun.
yeah, it was really, really amazing. Thank you.
Hopefully, people will use GeneLab now and come up
Yeah,
with interesting ideas. Great.
sure.
Thanks.
Thank you so much.
Okay, thanks. Yeah. All right. Bye.
Bye.